Yeah, thanks, Vital. It's a great pleasure to see you again, and thank you very much for the
Yeah.
invitation to this fantastic conference. So let me explain our recent progress on using polymeric nanomedicines for modulating or tuning immunity to fight cancer as well as other diseases. So we have been working with polymeric self-assembled structures to deliver different bioactive payloads um, inside the body with high precision. So what we are using is amphiphilic block copolymers that contains a hydrophilic segment. Usually this is polyethylene glycol and a drug loading block. And this drug loading block can be tuned to interact with the payload of interest. For example, small molecules such as anti-cancer drugs are usually hydrophobic, so we can make our drug loading block hydrophobic and eventually load them into a core shell nanostructure. But if our payload is, for example, a nucleic acid or a protein, we can change this drug loading segment to promote interactions, for example, with the negatively charged phosphate groups of the nucleic acids uh, with the drug loading block that is positively charged. So this process is done in buffer, so we can self-assemble the materials into these nanostructures that are around 10 to 100 nanometers. Yeah, so this is a core shell structure, so it's a polymeric micelle. The hydrophilic shell protects very effectively the payload in the core, and we can tune the drug release by, again, functionalizing the drug loading segment. In addition, we can put ligands on the surface to promote the interaction with tissues or cells directly inside the body. So with this formulation, we can modulate or overcome biological barriers in circulation. So once these particles are injected inside the body, we can tune the surface chemistry or the size to avoid distribution to measure elimination organs like the liver, the kidney, or spleen. For example, if we can make our particles bigger than 6 nanometers, we can avoid kidney filtration. Or if we make them hydrophilic and with a size below 100 nanometers, we can overcome the liver barrier. Additionally, this formulation has significant advantages for targeting solid tumors. They accumulate in cancer by what is called the enhanced permeability and retention effect, or EPR effect in short. So this is the increased leakiness of vascular tissues in tumors, so that macromolecules or nanoparticles can have enhanced access into the malignant tissue. So we have engineered our systems to have a enhanced penetration and ability to overcome the barriers that are also present in tumor tissues. For example, we can have a lot of fibrotic uh, uh, extracellular matrix surrounding the cancer cells, or we can have stromal cells like fibroblasts sitting between our blood supply and our target cancer cells. So we have designed systems that can overcome these barriers effectively. So for example, we can tune the size of the polymeric nanomedicines between 10 to 100 nanometers to overcome the fibrotic stroma. So in this case, it's an intravital microscopy in living animals. This is a pancreatic cancer, and we are seeing the flow of the nanomedicines into the tumor. So in green is a nanomedicine that is 30 nanometer. In red is a nanomedicine that is 70 nanometer. So as you can see in this movie, only the particle with 30 nanometer is able to penetrate deeply inside the tumor mass. We can also modulate the blood circulation of the nanomedicines. So if we can make it long circulating, we can take advantage of the dynamic changes of the vasculature of tumors. So the vasculature of tumor is not constant, so it's always changing, and there are fluctuations in the interstitial fluid pressure and the pressure in the blood compartment. So this allows for the extravasation in what we call vascular burst. So there are these discrete vascular points that are open and allow for the penetration of the nanomedicines into the tissue. We can further introduce ligands on the surface of the nanomedicines, and this allows for selective cell targeting, particularly if we are targeting the endothelial cells of blood vessels. We can promote the extravasation by facilitating transcytosis. Yeah, so for example, here we're using cyclic RCD in green, and this allows for penetration of the nanomedicine directly into brain tumors. If we use a control peptide that doesn't target the endothelial cells, it's not able to accumulate in brain cancer. So with these technologies, we were very successful in preclinical models, and because of this, we moved some of these formulations together with Professor Kazunori Kataoka, now at the Innovation Center of Nanomedicine, to test the possibility to treat human patients. So we developed several formulations, including anti-cancer drugs. Nanoplatin contains cisplatin, which is a platinum complex that binds to DNA and promotes cancer cell death. And epirubicin, that is an compound that it's a derivative of doxorubicin, and dahaplatin, which is another platinum drug comparable to cisplatin. So we reached advanced clinical trials. We were able to reach phase 3 clinical studies in patients with pancreatic cancer, and other studies like head and neck cancer into phase 1 and 2 in USA, Europe, and Asia. So these clinical trials indicate that the nanomedicines that we are making are very safe, with very reduced side effects. But unfortunately, the clinical trials also show that the efficacy was limited. And in a retrospective study, we and other groups have identified that the main limitation of targeting cancer cells in human patients is the variability of the EPR effect. So we have a variability between patients, and we have also variabilities within a same patient. For example, this study where they check for the distribution of liposomes in a patient that has a metastasis, the researchers observed that some metastases have a tumor accumulation, but other metastases show very low accumulation of the liposomes. So this heterogeneity is limiting the overall efficacy of nanomedicine. So we also noticed that in many cases, the cancer cells are surrounded by fibrotic tissue, or there is limited blood flow into the tumor area. So this, for a nanomedicine that wants to deliver an anti-cancer drug, is a very severe limitation. So we want to reach the cancer cells to deliver drugs that will make these cancer cells die. So instead of delivering this type of payloads, we shifted our attention to drugs that were involved in activating the immune system, so that we can promote the activity of these immune cells that are already infiltrating inside the tumor mass, so that we can promote their ability to kill the cancer cells. So one of such molecules are cytokines. Cytokines can activate the whole anti-tumor immunity cycle. But the problem is that when we inject a free cytokine directly into the bloodstream or systemically, they will cause systemic immune activation. So there will be an inflammatory response throughout the tissue. So this can lead to organ damage. And in addition, if we keep injecting the cytokine, the body will trigger counter regulatory immune suppressive signals. Yeah, so this will make the anti-tumor effect to be dampened. So we decided to put cytokines into our polymeric nanomedicines, and these cytokines are completely masked by the polymer, so they are inactive. And we provide the pH sensitivity to this formulation so that when they get to an acidic pH, the polymers are removed and we can recover the cytokine in the tumor tissues. So we focus on pH because pH is a hallmark. Yeah, tumor acidity is a hallmark in uh, not only in preclinical models but also in human tumors. For example, here is a patient with a liver cancer, and by pH mapping with MRI, you can see the pH is 6.2. Here is a breast cancer, the pH is around 6.3, 6.5. So we can make our system to sense this pH difference and activate the cytokine release. So this is the formulation we pursue. So it's again, it's a blocopolymer that we just mix with the cytokine, and this will associate with the protein to form a polymeric micelle that is stable at pH 7.4. That is the physiological pH. So the chemical structure of the polymer is the following. So we have the polyethylene glycol with a molecular weight of 10,000. So this provides effective shielding of the cytokine in the core, and a polylysine block. So this is a biodegradable, biocompatible polymer that has a positive charge for interacting with a negative charge in the cytokine, and a maleic anhydride moiety, which can bind with the amino groups in the polymers and also with the amino groups in the cytokine. So eventually, the core of the polymeric nanomedicine is cross-linked by pH-sensitive bonds that are very stable at pH 7.4. But once this particle enters a tissue that the pH is below 7, we can trigger the dissociation of the polymers and the release of the cytokine in the tumor's microenvironment. So to prove this concept, we focus on interleukin 12, which is extremely toxic, but has a very powerful anti-tumor activity. So if we can deliver and activate cytokine, the cytokine in the tumor tissue, we will be able to improve the efficacy as well as expand the therapeutic window. So the formulation is quite straightforward. We just mix the cytokine. Here is in the GPC. We can see the peak of the cytokine. Just mixing with the polymer, we have the formation of the particle. Some free cytokine remains. We eliminate this by ultrafiltration, and we have very nice particles of around 40 nanometers with one cytokine loaded per particle. Yeah, these particles are very pH sensitive, so they are stable at pH 7.4, but once we reach pH lower than 7, we can release the free cytokine. So we measure this by fluorescence correlation spectroscopy, and we follow the diffusion coefficient of the particle. And once the diffusion coefficient becomes larger, it means we are releasing the free cytokine. And this free cytokine is fully active, so it has a very strong ability to induce interferon gamma from immune cells comparable to the native protein. So the most interesting points happen in vivo. So we are having this cytokine injected systemically 2 times. So the first time we inject, we have a massive peak of interferon gamma in plasma. The second time we inject, the interferon gamma peak is much less, but we have a increase in interleukin 10. This means the body is trying to protect itself by producing an anti-inflammatory cytokine. So if we see the free cytokine in blood, is very much inflammatory, inducing different types of pro-inflammatory cytokines. However, inside the tumor, there is no inflammation going on. So there is no anti-tumor immunity being activated. On the other hand, if we use the nanoparticle, we have now no activation in the bloodstream, but selective activation inside the tumor tissue. So with this, we tested several tumor models. For example, we have injected this nanoparticle loading IL-12 into metastatic immunosuppressive breast tumor model. So we did this by inoculating the tumors into the mammary fat of the mouse. Then we remove the tumor at this time point. And in this condition, the mouse already have developed metastasis. Then we treat with the nano IL-12 or with the free IL-12 and see the anti-tumor activity. So this is histology at endpoint. The combination of nanoparticles with immune checkpoint inhibitors allow for complete eradication of the tumor mass. And then we have a complete survival of the animals in this animal experiment. So one limitation of this approach is that proteins are quite expensive, and it's maybe difficult to for startups to formulate cytokines and other protein therapeutics into clinical drugs. So we then shifted our attention to messenger RNA. So messenger RNA can reduce the cost of GMP protein manufacturing and provide the in situ production of therapeutic proteins in a safe manner compared to other approaches like plasmid DNA. So we can then engineer our system. For example, we can use PEG polylysine to interact with nucleic acids like messenger RNA to form core shell nanoparticles for delivering to tumor tissues. Yeah, so we have shown that this system is quite effective to promote intracellular delivery of messenger RNA, as well as reducing the immunogenicity of the messenger RNA in this intracellular delivery. So the process is just by mixing the glycopolymer with the messenger RNA, and we can do this in buffer without any addition of organic solvents or no microfluidic machines. So the size distribution is quite homogeneous by DLS and also by TEM. And because of the high PEG shielding of the micelle, we can prevent interaction with the toll-like receptors. So this can evade inflammatory responses of messenger RNA. And in addition, we can engineer the polycation segment to provide buffering amino units to promote endosomal escape at endosomal pH 5.5. So we can still use the pH-sensitive cross-linking for the messenger RNA system. So for example, here we have changed the maleic anhydride moiety to a system that can sense endosomal pH. So we can just mix the messenger RNA with the glycopolymer in buffer, incubate at pH 7.4, and we can have a cross-link core with a pH-sensitive bond that will cleave in intracellular endosomal pH. So this particle is around 50 nanometers, and if we inject directly into the tumors, we have a very high production of proteins. This is observed by luminescence using a firefly luciferase, and we can achieve much higher signal than commercial transfection reagent like polyethylenimine. So, but if we inject this particle systemically, then the particle will distribute to different tissues inside the body, and it will produce the proteins not only in the tumor tissues, but also in off-target tissues like, for example, the liver or lungs. Yeah. So if this is a cytokine, the production of these cytokines in off-target tissues can lead to unbearable side effects. So to give one extra layer of safety, we design a photoactivatable polymeric micelles that can only produce the therapeutic proteins after light irradiation. So we do this by integrating a photosensitizer agent into the polymer. So this is again polyethylene glycol and polylysine. It's comparable backbone that we use for the IL-12, but instead of having the cross-linking moiety, we introduce this photosensitizer molecule. So this can be activated with a near-infrared light and produce a photothermal effect as well as a reactive oxygen species. So we mix this polymer with IL-2 messenger RNA, and this positive charge in the lysine will complex with the negative charges of the messenger RNA, and this planar structure of the photosensitizer will make pi-pi stacking with the basis of messenger RNA. So this stabilizes the structure against ion competition and other biological signals that may want to disrupt the polymeric structure. So this micelle, when reaches the tumor, it enters cancer cells and it goes into the endosomes. And if we give the light irradiation, we can now make this photosensitizer create ROS and photothermal effect in the endosomal compartment. So the endosomal compartment will be disrupted, and we can get the release of messenger RNA directly in the cancer cells. But if we don't give light, for example, if we don't give light in healthy tissues, then there will be no endosomal escape, and there will be no protein translation in healthy tissues. So then we can have a selective production of IL-2 in these tissues to activate the immune system. So we test this idea in the lung metastasis model. So we have created the lung metastasis in these animals. We inject our IL-2 messenger RNA made with this polymer, and then we give light irradiation to the lung of the animals. So if we don't give any light, we have very low signal of interleukin-2 being produced in the metastasis. But if we give light, we can now increase these levels more than 3-fold. And this treatment allows us to eradicate 4 out of 6 of the tumors. Yeah. So we can cure 4 animals out of 6. So besides cancer, IL-2 has also the possibility to be useful for immunoregulating therapies. So at high dose, IL-2 can activate natural killer cells or cytotoxic CD8 positive cells and increase their activity and proliferation. But in low dose, the signal is quite opposite. Actually, the IL-2 can expand and proliferate immunoregulatory T cells. So we can make a low dose IL-2 to promote this type of T cells to create immunoregulatory treatments. However, these creating these Tregs is very dependent on the inflammatory conditions of the tissue. So if we can reduce the immune response of our delivery system, we could selectively promote the proliferation of regulatory T cells. So we did this by, again, modifying the polyethylene glycol polylysine, and in this case, we introduced curcumin. So curcumin is a ROS scavenger. So when ROS are being produced in the cells, the curcumin will uh, eliminate these reactive oxygen species. So we just mix with the messenger, make the micelle, and check for the inflammatory reaction. So without the curcumin, we have some inflammatory signals. When we put the curcumin in the polymer, there is no inflammatory signal created with this polymer. And we can have a promoted interleukin-2 production in tissues. So we test this in a model of ulcerative colitis. The polymer with curcumin can now have a very strong effect on the ulcerative colitis by creating more regulatory T cells, as well as um, reducing the amount of cytotoxic T cells. So this is my last slide, but most important, I would like to thank the members of my laboratory who did all this tough work that I present today, my collaborators in Japan, my collaborators overseas, funding agencies, and industrial collaborators that make all this possible. Thank you.
Okay, thank you very much, Horacio.
Thank you.
There's a